Protein denaturation. Let's talk about denaturation here first. What do we mean by denaturation? So you go from the native protein, so you think native or normal protein. What we mean by that is normal structure. This is how it's supposed to be. So this is active or working. It's working the way it's supposed to. It's active. There's a number of things we can do to unfold this, unravel it. Engineering and design, but also in biochemistry, we talk about form follows function, or function follows form. If it doesn't have the right form, it's not going to have the right function. So if you disrupt this normal native folding state and you kind of unravel it, it's denatured. So this denatured is not normal. We lose, so it's not the normal structure. So it says loss of structure. What we're losing is the normal structure. So it also loses a function. Often it also loses its solubility. Talk about function first. So just say here again, not working. What do we mean by solubility? Well, remember in the interior, right, we had hydrophobic regions. When you unravel it like this, it exposes these hydrophobic regions, and it is no longer like a nice happy fatty acid micelle where all the hydrophobic tails are on the inside. You have them all sticking out all over the place, and it just doesn't interact with water properly. You can also form cross-links with other proteins. So you're never working with one protein in isolation. You have other proteins. And so what happens is you get a precipitation from cross-links between different proteins. They don't show another unfolded protein here, but we can put another protein in here like this, maybe. Okay, and it'll form cross-links with this one that's also unraveled. We can put a third one in here, maybe. That's also unraveled, and you get this big unraveled mess of proteins that comes out of solution or precipitates. Now, in very rare cases, once the protein is denatured like this, can it be reformed? And I'll talk about that a little bit at the end of this lecture. You go from denatured to native. This is very rare. Usually what happens is once you denature it, it's irreversible. You cannot go back. So it's denatured and it's done, it's dead, and that's the end of that protein's life. How do proteins get denatured? Well, we already mentioned this a little bit. We talked about heat, right? Heat will disrupt hydrogen bonds. Right? So you can think of denaturation in a mild form. Fever. Right? Fever is the body's response to kind of ramp up some chemical reactions and fight infection, but if the fever gets too high, the body basically cooks itself and denatures its proteins. Speaking of cooking, if you ever, I don't know, if you're cooking a steak, if you're cooking a steak or maybe boiling an egg, okay, the white of the egg is protein essentially called ovalbumin. And when you boil that egg, that ovalbumin gets denatured and it comes out of solution, right? It was a liquid, it becomes a solid, it precipitates. And so you go from, say here, egg white, that's all gooey, kind of gelatinous, it's a liquid, and you heat it up, and you still have egg white, but now it's really white, right? Because it's a solid, it's precipitated, right? So that's what happens with heat. You're disrupting these hydrogen bonds. Detergents. It says here detergents, but it could also be soaps. Right? And one of the ways we fight COVID-19 is by washing our hands, and that disrupts the viral coat of the proteins of the coronavirus and destroys it. So what we're doing here, remember, soaps have this long chain. And basically what we're doing is we're sticking that long chain in here and disrupting the London dispersion forces or van der Waals forces. So disrupts the van der Waals forces, the London dispersion forces, or hydrophobic interactions. This is another term you hear, hydrophobic interactions. Agitation? What do I mean by agitation? Basically just mixing or beating it. Mixing or beating. Again, you can. I'll go back to eggs. Right? You could take an egg white, separate the white from the yolk. I don't know if you've ever done this before. And you can take your blender and beat it up, and it will denature, and you get something called, what is it? Is that how you spell meringue? Or is that merengue? I don't know. But you get, you take egg whites and you beat it up, and the proteins get denatured, and you form a meringue. Disulfide reagents we talked about. You disrupt the disulfide bonds. I'll put it here. Right, so you oxidize, I'm sorry, you reduce these disulfide bonds. So we can put disulfide reagents here. I just give you a couple that you might, I don't know if you ever come across. One is something called mercaptoethanol or ethane thiol. It's just essentially ethanol, right? Ethanol would have an ethyl group and alcohol. Well, this is SH. Another one is called DTT. Now, DDT, I'm going to put it down here. DDT is a banned insecticide. I'm writing this the day after Earth Day. The first Earth Day in the environmental movement, 1970s, was really kind of started a little bit before there with a book called Silent Spring by Rachel Carter. Silent Spring, which talked about how this insecticide was killing all the birds. So in honor of Earth Day, I'll just mention the book that she wrote, Silent Spring, has to do with this insecticide DDT. I'm not talking about that. This is DTT. It stands for dithiothriotol. That's also sometimes called Cleland's reagent. I'm not going to draw the structure, but essentially it is also a thiol reagent. And the reason it's called Cleland's reagent is the chemist who came up with this, a fellow by the name of Maurice Cleland, was also a professor at UW-Madison, so Wisconsin history there. So anyway, thiol reagents. Basically what you're doing is, instead of forming a thiol between, or disulfide between two different cysteines, you're forming the thiol between this ethane thiol, so a small molecule with this thiol, and not between these two. And then once this is broken, the two parts can kind of unravel. All right, lastly, acid or base. Acid or base denaturation. Again, you could try this with, you could try this with milk. When milk goes sour, right, what's happening is there's bacteria in the milk that are oxidizing the sugars, and the sugars get oxidized to acids, and the acids denature the milk proteins, and you get this clumpy soured milk, right? If you've ever poured milk into a glass and it came out in clumps and it smelled really bad, that's essentially what's happening. The pH drops, and you can do this with either an acid or a base, but what you're doing here is breaking what they call the electrostatic interactions, or again we call those salt bridges. So acid or base. If we add acid, what we're doing is putting a hydrogen on this carboxylate. Well, now it's not charged anymore, so we lose that electrostatic interaction. If we have a base, what do bases do? Bases, right, this OH is a base. It's going to remove one of these hydrogens to form water, and you'll go from NH3 with the plus charge to just NH2. You'll lose the plus charge. Again, the plus charge there is if you eliminate the plus charge by adding a base, now the carboxylate doesn't have anything to bind to, and the protein can unravel. So know these different side chain interactions and know how to disrupt each one of them. A little more vocabulary here. You might have heard of some of these words, some of these terms before. You might have heard of something called proproteins. So there's pre-proteins, proproteins, apoholo, something called a prosthetic group. Proteins, again, we're going to go from DNA is converted in the nucleus. So this is nucleus. In the nucleus of cells, transcription. So it's transcribed a messenger RNA. Messenger RNA then leaves the nucleus. It leaves the nucleus, goes to the rough endoplasmic reticulum. It's outside of the nucleus now. Bonds to something called a ribosome. And then the ribosome translates that messenger RNA code to protein. So fine, you've got a protein now. So what you're doing is you're taking a bunch of individual amino acids and converting them into a string of pearls, right? A bunch of different amino acids held together. That's a primary structure. That folds up to give you the protein. But we're not done there. Oftentimes this protein is then what we call a post-translational modification. And there's a number of things that we can do. We could add carbohydrate, right? So we could get a glycoprotein. Talked about the role of carbohydrates decorating the surface of proteins, glycoprotein. You could add lipid. You get a lipoprotein. Could add something called a prosthetic group. Talk about what that is in a moment. And we could also have what we call a proteolytic processing or cleavage. And so basically what we're going to do, this is cleavage. Basically what we're going to do is chop this protein up into smaller pieces. So what happens a lot is, now the insulin is a good example. This is pre-insulin. So insulin is made inside the pancreas cells, and it needs to get outside, right? It's a peptide hormone, it needs to circulate in the blood. The signal peptide tells the cell, hey, it's like a zip code or something. It tells the cell what to do with it. So it signals a peptide. I just say it tells cell to export this protein outside of cell. So, okay, clip that off. Now we have proinsulin. So it was pre-proinsulin. A pre-protein, right, has a pre-sequence. That tells the cell where the protein should go. Some of these pre-sequences, again, will tell the cell to export it. Some tell them, oh, maybe it's a nuclear protein, so it has a nuclear targeting signal peptide that tells the cell, hey, this protein, remember, is made outside of the nucleus. It's got to go back inside. Maybe it's involved in DNA repair or replication. Maybe it's destined for some other intracellular organelle like the mitochondria or the lysosome or the Golgi apparatus or something like that. And then there's proprotein. So we've got this proprotein, and what we're going to do is we're going to, again, post-translational modification. This is a proteolytic, both of these, the signal peptides cleaved off, these bonds are broken. Both of these are proteolytic cleavages. Proteolytic cleavage. We're going to break the peptide bonds here. Sorry, first here, and then here and here to get the native, or maybe what we will call mature form of the insulin. So a lot of proteins are made this way. They're made initially as a proprotein. Both of the pre-protein and proprotein are inactive. Right? So proprotein is a way of essentially storing inactive protein until it's needed. And there's reasons why cells would like to do that, like to store protein in an inactive form, and we can talk more about that in a little bit. Then we have apoproteins. A good example of this would be the globin chains of hemoglobin. All right. So apo, also inactive. Essentially, an apoprotein, not all proteins are going to work right away all by themselves. Sometimes they need, if you wish, helper molecules or something. That helper molecule is a prosthetic group. So a prosthetic group is a non-protein molecule that's required for function. Required for function. So what we're going to do is we're going to take an apoprotein, we're going to add a prosthetic group, and then we get the holoprotein. So the holoprotein is apoprotein plus the prosthetic group. So I was talking about hemoglobin. The apoprotein would be the globin chains. So that's the protein part. There's actually two different kinds. There's alpha and beta chains for hemoglobin. The prosthetic group is the heme. It's a non-protein molecule called a heme. That's the prosthetic group. It gets covalently bound to, so incorporated into the protein, and then we have the holoprotein. And now this is finally active, the working functional protein is the holoprotein. Okay, so as I mentioned before, here's hemoglobin. We'll talk about it in a moment. Myoglobin is, I just say here, this is the oxygen carrying O2 carrying protein or enzyme. Well, it's really a protein in muscle. Hemoglobin, its function is the O2 carrier in blood. Okay. So the heme is this black atoms here. In the center of this heme, we have an iron atom, also required for function. It's supposed to be Fe for iron, Fe plus 3. This is called a cofactor. The heme is organic. Okay. The cofactor is inorganic. That's the main difference between a cofactor and a prosthetic group. One of the main, another main differences are the prosthetic groups are covalently bound to, it doesn't show it here, but it's covalently bound to the protein. Cofactors, not necessarily. So, and then the cofactor would be inorganic. Here we see hemoglobin. It consists of, do they color code them? No, they just say red and blue. So we have 2 alpha chains and 2 beta, so find the alpha here will be in red and 2 beta chains in blue. So this has a quaternary structure. Right? You can just see where we can find. Can't really find a good end here to work with, but maybe here is a amino terminus, so you can follow this around. You have some alpha helix, and it goes around here, some more alpha helix, and so that's the secondary structures. The alpha helices combine together to form a tertiary structure. Individual polypeptide chains have a primary, secondary, and tertiary structures. They combine to form this quaternary structure. So this is the quaternary structure of hemoglobin. We say it's an alpha 2 beta 2 tetramer, right? 2 alpha chains, 2 beta chains, and then those would be the globin chains are the apoprotein. Once we put the heme inside, right, then that's a prosthetic group. Then it is a functional holoprotein. So again, apoprotein plus the heme, which is a prosthetic group, gives us the holoprotein. Alright, so that will do it for this unit on proteins. I strongly encourage you to watch that other video entitled The Protein Folding Problem. It turns out that with today's modern computers and the fast computers we have now, and all the other proteins I've been able to figure out what their structures are through X-ray crystallography, that database has allowed us to produce algorithms that will, without having to grow those protein crystals you saw, and then collect a diffraction pattern and use that for deducing what the electron density is and fitting the amino acid sequence into the electron density. We can basically just go from the amino acid sequence using a computer now, calculate what the protein structure is going to look like. Not 100% of the time, and not for all proteins, but for a good portion of them. And the other thing I'd like to point out is that the first protein that they show there, if you look at that video where they show the protein folding, that protein is actually the protein I worked on in graduate school. And it's a small protein. It's only 76 amino acids, so it barely makes it in terms of the size for proteins. Most proteins are much, much larger, say 10 times larger than that. So, hundreds of amino acids, not just 76. But the neat thing about it is you can denature it, so you can precipitate it in acid, and then you can wash away the acid and it will refold. And so it's a good example for protein folding, and that's what that video shows. By the way, the name of that protein is, I wrote it down here, so we can go from, I just say ubiquitin. It can be denatured, so you could add HCl. You could remove the HCl, so just say neutralize, and you get the folded protein back again. Folded protein back again. So ubiquitin comes from, ubiquitin is derived from the name ubiquitous. It is a very ancient protein in an evolutionary sense. In what I mean by that, it's highly evolutionary conserved, about the most highly conserved protein around. Histones, these are proteins that are involved in packaging DNA into our nucleus. They're also highly conserved, but ubiquitin has only 3 amino acid differences between the ubiquitin that's in your cells and the ubiquitin that is in a fungus like yeast. So a lowly primitive organism like yeast has essentially the same exact protein as you do. So be sure to watch that TED Talk on protein folding.